Hello, everyone. Now let's study the chapter 7, microbial metabolism. Now let's work on the section 1. So the relationship among metabolism, catabolism, and anabolism. The metabolism means all the chemical reactions and the physical workings of the cell, because the cell can be considered as very small, tiny chemical factories. So every day, millions chemical reactions happens in this small factories. So the metabolism contains anabolism and catabolism. Anabolism means all the process that the cell, so all the process that the cell have done to make cellular materials for the cell, to make to build the cell, to make the cellular memories, to make all the cells molecules and structures. So that means the cell can use the raw materials, so the small molecules to make the huge molecules such as carbohydrates, lipids, and proteins. Yes, so the cell uses small molecules to make the macromolecules. Catabolism is inverse, so the cell will break the bonds of the macromolecules to small molecules and release energy. So the cell can use energy for their life. So as what we can see in this diagram, so the glucose can be breakdown, can be catabolized to through the glycolysis, Krebs cycles, and fermentation to the small molecules and release energy. So the small molecules such as pyruvate and acetyl-CoA, so they can be used by the anabolism to make amino acid, sugar, and nucleic acid and fatty acid. So so those would be the anabolism. So the cell can use these macromolecules to build the cell itself, such as the proteins, the membranes, and nucleic acid. So all the metabolism is regulated through the enzyme. So the enzyme as catalyst. Catalyst means so they are the specific chemicals or the molecules that can regulate the process of chemical reactions. So they can increase in most of cases. So catalytic. So the catalyst increase the speed of chemical reactions. So they don't become a part of the products. So they are not consumed in the process. So they don't. So the catalyst doesn't create a new reaction. So they just so they are just involved in the reaction. However, they don't create a new reaction. So the characteristics of enzyme is so enzyme are the proteins, okay, are the proteins. Also, they also need cofactors. Uh, so the major part is the protein. So they can regulate the chemical reactions speed. So the enzymes have a specific shape and function. Okay, so the function is determined by the specific shape. Okay, so enzyme have active site and regulated site. Active site is for binding uh, target substrates. So the enzyme is larger than the substrate because because they have to larger surface for binding the substrate. That means they have to be larger than the substrate. So so they have the so they have the so they have the active site that has structure close to the substrate, but the enzyme itself doesn't become part of the reaction or the products. So enzyme itself will not be changed by the reaction. So the enzyme can be reused again, so can be recycled because they are not changed. So if they are changed by the reaction, so they cannot be recycled or reused again. Uh, so the enzyme actually the enzyme has very high efficiency uh, for the for increasing chemical reactions. So that's why so the cell doesn't need a high concentration enzyme. So they just so the cells only need a small a little bit of enzyme for all the reactions. So enzyme is also regulated by the temperature and pH. So so that's why uh, so that's why all the animals and humans have their own body temperature, right? Because so the body temperature actually is the optimal temperature for the enzyme. So the enzyme can uh, perform their highest efficiency at that temperature. So it can be regulated the feedback and genetically can be regulated by the genetic mechanism. So we talk about this later. So how do enzyme work? There's the enzyme. So enzymes have specific sites. So they have the active sites for substrate to bind. Okay. So so they are large. So they are larger than the substrate. So so first of all, so the unique active site of the enzyme will bind the substrate, and then so the enzyme can change the substrate, and then so for example, to sanitize a specific molecule, so the enzyme can bind two substance two substrate for that molecule, and then so the enzyme can make them closer together to increase the chance for the reaction. For example, in this image, we can see, so this is enzyme, okay, this is enzyme. So the red one is the cofactor. Cofactor normally is metal iron, okay. So the cofactor plus the enzyme is called the holoenzyme. So the enzyme itself is called the apoenzyme. Sometimes the cofactor can be a small organic molecule, so it is called a coenzyme. Okay, coenzyme. So the enzyme itself is the protein, okay? So it is called apple enzyme. Apple enzyme. So the apple enzyme plus the cofactor is called hollow enzyme. Cofactor. So either cofactor is the organic molecule is called coenzyme, or the inorganic elements such as metal, ions. So this is called a cofactor. Okay, we can directly call this one as cofactor. So the most important structure of the enzyme is the active site. So the active site is the major part for the enzyme to bind the substrate. So this is the structure of the active site is very specific. So it's real specific to fit the substrate. So this is also described as local and key fit because it's exactly fit the substrate. So once the enzyme bind the substrate, so a chemical bond can form between the substrate and enzyme. However, the bond is weak and easily reversible. Okay, so such as the hydrogen bond. Okay, it's not the strong bond. It's not the iron bond. Okay. So once the enzyme bind with the substrate, uh, so the enzyme can change the substrate, either change the substrate structure or uh, or make close of the substrates together for the reactions. So this process have to be aided by the cofactor. Once the reaction is done, so the product can be released. So enzyme is free, and so the enzyme will be reused, can bind with the other substrate for the reaction. So as what we can see in this diagram, so the enzyme have its specific active site. So the structure of active site is exactly fit the substrate. So this is called lock and key fit. Lock and key fit. So the active site can only find one specific substrate, so not others. Okay, the others substrate have their specific structure, so they cannot fit the active site. So once enzyme bind the substrate, so the reaction can happen. For example, for degrade this molecule, so the enzyme can change the structure of the substrate, can degrade this molecule to two products. Then the products will be released, so the enzyme is free. Enzyme can can bind another substrate for the reaction. So the cofactor actually, so the microorganism doesn't need high high uh, high volume of the cofactors, so they just need a small volume of the cofactors. So normally the cofactors are the trace elements. For example, the iron, okay, copper, magnesium, and magnesium, zinc, cobalt, selenium. Okay, so so the percentage of this element actually in the cells is very, very low. So this is called trace elements. So the cofactors are involved in the enzyme activities. Uh, so they can bring the active site and the substrate close together, so they can directly so they can be directly involved in the chemical reactions to increase the speed of chemical reactions. So the coenzyme actually is one type of cofactor. However, so they are the organic compounds. So they are not the elements. Okay, so they are organic compounds. So they can bind with enzymes. So their function is to remove a chemical group of the substrates and or added to the substrate molecules. They can also carry or transfer hydrogen atoms, electrons, carbon dioxide, or the amino groups. So many coenzymes are derived from vitamins. So based on the active site and chemical reactions and the substrate, we can name the enzymes. So we can add the prefix of the stem words. We also can add endings of the ASE to the words for the names. So the common name is generally based on the biochemical actions. Okay, for example, carbohydrates, and the proteins. Proteins means that enzyme work on the protein, right? To degrade the protein. Carbohydrates means enzyme to degrade the carbohydrates. Lipids is the enzyme to degrade the lipid. Polymerase means make the polymer. Okay. So as what we can see in this diagram, so the enzyme we can divide the enzyme based on their functions. So one is the constitutive enzyme, another is regulated enzyme. Constitutive enzyme means the enzyme numbers are not changed during the chemical reaction. However, the regulated enzyme means the enzyme's number can be increased, can be either increased or decreased based on the chemical reactions. So if there are more substrate for the chemical reactions, so the regulated enzyme number can be increased to increase the chemical reactions and produce more products. So once the chemical reactions is done, so the enzyme numbers can be returned to the baseline level. Okay, can decrease again. So this is regulated enzyme. So the enzyme can be regulated. Okay. So they can be regulated by the cell's environment, such as the temperature, pH, and osmotic pressures. Okay. So even in the normal conditions, so the enzyme can also be changed or regulated. So this is called so this means the enzyme actually is very labile. Okay, labile. So the enzyme can also be damaged by certain conditions. So we call this as denaturation. Denaturation. So that's mean for the denaturation actually, so the enzyme structures are changed. Okay, so the because enzyme actually is the protein, so the protein has specific tertia or the uh, or and the coordinates structure. Okay, all these complicated structure actually are maintained by the hydrogen bonds. Hydrogen bonds are the weak bonds. Okay, so they can be damaged by certain conditions. So if the hydrogen bonds are broken, so the proteins or the enzyme structure can be changed. So the denaturation will happen. So the structure is changed, so it will cause distortion of the enzyme shape. Okay. So once the enzyme structure is changed, so the active site structure is changed, so the enzyme cannot bind with the substrates. So there are some chemicals that can inhibit enzyme activities. So based on the function of these inhibitors, we can divide those inhibitors to two categories. So the first one is the competitive inhibition. Competitive inhibition means these chemicals can directly because these chemicals has a similar structure as substrates, so they can directly bind the active site of the enzyme. So they can mimic occupied the active band. Okay, so once they bind with the active site, so they can prevent the real substrate from binding. Okay, so that's why it is called competitive, because so these chemicals compete with real substrates. So if this happens, so obviously, so the chemical reaction speed will be decreased, right? So the second is non-competitive inhibition. So the inhibitors actually don't have the uh, similar structure as the real substrate. However, they can bind to the regulatory site of the enzyme, because the enzymes have two specific sites. So the first is the active site. Active site is used to bind with the substrate. Another is the regulatory site. So the no-competitive inhibitor can bind with the regulatory site. Once they bind with the regulatory site, uh, so they can change the enzyme structures, or um, or they can, make the enzyme very low efficiency, very low efficient to perform the enzyme functions. So they can slow down the enzyme activities. Okay, so the chemical reaction speed is decreased, and yeah, so this is non-competitive inhibition. So as what we can see in this diagram, so this is competitive inhibition. So inhibitor has similar structure as the real substrate, so they can compete so they can compete each others to bind with the active site and decrease reaction chemical reaction speed. So for the non-competitive inhibition, so the inhibitors doesn't inhibitors don't bind with active site, so they bind to the regulative site. So the inhibitors can be the products of the chemical reaction, so they can go back to bind with the regulatory site of the enzyme to decrease this chemical reaction. So actually, this is called a negative feedback pathway, right? So this actually, so non-competitive inhibition is widely exists in the cell for the negative feedback pathway. Now let's talk about the control on the enzyme. So the enzyme actually is made. Enzyme is a protein, so it is made by the by the rough, yeah, by the ribosome, right? However, the actually, so the regulation is maintained by the DNA. So the once because the DNA contains all the information for all the enzymes. So if the cells need certain enzymes, so the DNA can transcribe those enzyme information to make RNA, and the RNA can reach to the ribosome to produce the protein. So actually, so the genetic actually the DNA, so the genetic apparatus, so they are responsible for the enzyme regulation. Or they can make less enzyme, so they can repress enzyme numbers to decrease certain reactions. So since this is actually, so this is a very long pathway, right? So this pathway is involved in the transcription, translation. So that means so it takes a long time to go through this pathway. But this pathway is very strong, okay, and if this pathway is initiated, so the effect is long, okay, so it can continue produce more enzyme or continue decrease the enzyme. So the enzyme induction, so that means make more enzyme, okay. So if for example, if so there are more substrates, so the cell need to increase the chemical reactions, so the cell will make more enzyme. So this is called enzyme induction. So for example, as what we can see in this diagram, okay. So once the DNA make RNA that make the protein becomes the enzyme. So enzyme will mediate the chemical reactions and make the products. So the products can go back and bind with the DNA to decrease to inhibit the DNA to transcribe the information to make the enzyme. So this is called enzyme repression, okay? We can see, so this is also an example of the negative feedback pathway, right? Now let's talk about the E. coli, okay? So the enzyme induction in E. coli. So the E. coli, so if we inoculate the E. coli into the medium, so if that medium contains lactose, so the E. coli will produce lactase uh, to degrade the lactose to the glucose and galactose. Okay. However, if the medium contains the sucrose, so E. coli will synthesize the so the E. coli will not synthesize lactase, okay, but they will synthesize the sucrose. Then so they can use the sucrose to degrade the sucrase to degrade the sucrose. So this is so that means the E. coli can make a different enzyme based on a different substrate. Okay, so this is a strategy for the E. coli to adapt the environment and so don't waste the energy, don't waste the material to make the wrong products. Now let's talk about the metabolic pathway. So the metabolism contains anabolism and catabolism. Actually, so the anabolism and catabolism is linked together. So the products of catabolism can be used by the anabolism to build new products. Also, the energy released from catabolism can be used by the anabolism to build more materials. Also, the products of anabolism can also be catalyzed by the catabolism to provide new small molecules for the anabolism. Actually, so they are linked together. So the pattern of the reaction could be the linear, okay? Could be the linear, okay, or the cycle or the branched. For example, the A can be catalyzed to B, B can be catalyzed to C, C can be so C can be synthesized. C can be synthesized to D. Okay, so this is linear. So the example would be the glycolysis. So the cycle, so the example for the cycle is the Krebs cycle. Okay, so the U can be made to V. Okay, V can be catalyzed to W. W can be catalyzed to X, and then to Z. Z can be made to V. Okay. So the branches means so there are two pathway. Okay. For example, N to O, N to P. Okay. Or like this way. So this is the conversion. So this is the diversion. Okay. So the reason to have the branches metabolism pathway is that actually so the microorganisms have been evoluted for million years. So they have to adapt to very tough environment, very tough conditions. So that's why so the microorganisms have the plan B for certain regulation, right? So if one pathway is inhibited or abolished, so they have another pathway to 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 still produce the products, right? For example, if this way doesn't work, so they still have the plan B this way for the metabolism, so they cannot die. So now let's work on this question. Which of the following mechanisms of the enzyme control blocks a reaction catalyzed by an enzyme by the binding of products to a regulatory site on the enzyme? So I give you 5 seconds so you can review the previous slides and give me the correct answer. So we can see the enzyme repression is not correct, okay? Competitive inhibition is not right because the competitive reaction bind with the active site. Enzyme induction, no. Enzyme induction means make more enzyme. So non-competitive, yeah, non-competitive is right because non-competitive inhibition is to bind with the regulatory site. So now let's work on the section 2. So the energy. So in all the living organisms, okay, so most of the living organisms actually use ATP as the as a transporter of the energy. Okay, so the energy actually is stored as ATP. So the ATP can provide our cells energy. However, the energy is from the chemical reactions. Okay, energy is produced from the chemical reaction. Actually, it's from the catabolism. So the energy actually is from is made from breaking the chemical bonds of the molecules. Okay. So based on the energies, we can classify as the exergonic reaction. Okay. So exergonic reaction means to break down the molecules and release energy. So makes the energy available for cellular work. So second is the endergonic reaction. So the endergonic action means, for example, for the anabolism. So the cell will make new molecules. So the making process need energy, right? So the energy is used by the cell to make new molecules. So the energy is added to the chemical bonds of the new products. So this is endergonic reaction. Actually, so the exergonic and the endergonic reactions are coupled together. Okay, are coupled together. So the cells have a specific enzyme to process the energy. Okay, either releasing energy or using energy. For example, so if the energy is released during exergonic, so the energy will be stored in the ATP, okay, and then ATP transfer energy to all the cellular organelles for all the cells work, okay. So ATP temporarily store and release energy, okay. So how the cell release energy, okay. So actually the most classic reaction that cell release energy is oxidative phosphorylation. Okay, so let's talk about the oxidation. Okay, so the oxidation means loss of the electron. So when a compound loses electron, so it is oxidized. So reversely, so the reduction means the gain of the electron. So once the compound gains electron, so this compound is reduced. So the reaction, chemical reactions of oxidation and reductions, so it is called a redox reaction. Okay, redox reaction. Actually, so the oxidation and reduction, so they are linked together. Okay, so they are not separated. Actually, they are linked together during the releasing of energy. So this is the redox pair. Okay, as we can see in this diagram. So this is redox pair. Okay, that means oxidation and reduction is linked together. For example, for the formation of the salt for the sodium chloride. So the sodium atom will lose one electron. So the sodium atom will be positive charge. Okay, so this is oxidized. So it becomes to one cation. So they will donate the loose electron to the chloride. So the chloride atom can gain one electron from sodium atom. So the chloride atom is reduced. So the chloride atom is negative charge. So the chloride becomes to anion. Okay. And then since this one is positive, this one is negative, so they can attract each others and bind each others to form sodium chloride. So this is the salt. So the redox reaction can be regulated by the enzyme. So the enzyme is called oxidoreductase. So this is the enzyme specifically regulates the redox reaction. So they can remove electron or add the electron. So they can remove electron for one substrate and add electron to another substrate. Okay. So the specific oxidoreductase used in living organism is so there are two specific enzymes. So one is the nicotinamide adenine dinucleotide. It is also called NAD. Okay. The second is the flavin adenine dinucleotide. So it is also called as the FAD. So those enzymes actually so they work as transferring of the electron. Okay, so they are the oxidoreductase. So the redox pair. Okay, so that means electron donor and electron acceptor actually, so they are linked together. Okay, to form redox reactions. So as what we can see in this diagram, so NAD can accept one electron from the hydrogen. Okay, so they can absorb one electron and one hydrogen atom from hydrogen from the hydrogen molecules and make another hydrogen atom a positive charge. Okay, so they can transfer. So once they once they absorb the electrons, so they are be so they can so once they absorb the hydrogen and the electrons, so they will be reduced. Okay, so this is reduced. So for example, they can be reduced. So originally, so this is oxide. So this is oxidized NAD. Okay, oxidized NAD can accept electron and hydrogen ion, so they can become to reduced NAD. Okay, and release hydrogen ion. So so once the electron is accepted by certain acceptors, uh, so the so the energy can be stored, can be captured to by the phosphorylation. Okay, phosphorylate. Okay, um, for example, for the ATP, ATP is a good example for explain the phosphorylation. For example, once the ADP, A means adenine, D means double, okay, P means phosphate. So that means this molecule has two phosphate, okay. So they can so it can accept another phosphate and form chemical bonds between the third phosphate, so they can store the energy in the third phosphate bond. Okay, so they can store the energy. So the electron, so the cylinder can generate the energy by handle the electron and the hydrogen ions. So actually, this is called cylinder respiration, or the electron transportation chain. Okay, so we talk about this later. Dehydrogenation. Okay, this means remove hydrogen. Okay, remove hydrogen. So we talk about this later. Now let's talk about the ADP. Okay, ATP. ATP. So it has three parts. Okay, it has three parts. So it has actually it has ribose of 5-carbon sugar. Okay, a five carbon sugar and the nitrogen base. Okay, nitrogen base actually is adenine. Okay, it has a chain of three phosphate group. Okay, so this is called TP. Okay, TP. Uh, so the phosphate group are very large. Okay, so they are negative charge. Uh, so they can so the three phosphate group can either lose one phosphate or gain one phosphate. When they lose one phosphate elements, so it will be double phosphate group. Okay, so this is called ADP. So once they lose the terminal phosphate group, so they will release the energy. Okay, so that is how the ATP store or release the energy. For as what we can see in this image. Okay, so this is adenine. Okay, this is adenine. So this is ribose. Okay, so this is ribose. This is adenine. So this is nitrogen base. Okay, so we can see the three phosphate group actually are linked with the ribose. So the terminal one can be either released or conjugated. Okay, so the energy can be released. So the third phosphate bond can be released, can release energy. Once the terminal phosphate is released, so this molecule is called ADP. Okay, so once the second phosphate is released, so this molecule is called AMP. So M means mono, so means one, just one. Okay. Actually, so the AMP cannot release energy anymore. Okay. However, AMP is widely used as secondary signaling messenger, right? So the recycling of the ATP actually is how the cells utilize and releasing the energy. Okay. So once ATP is made, okay, so the terminal phosphate uh, will store the energy. So once ATP is used, it's degraded. So the third phosphate is released, and so the energy is released from ATP for the cells to use. So for the microorganism, so actually, so the catabolism provide energy. Okay, that energy can be stored to the ADP to form ATP. Now let's work on this question. In a redox reaction, loss of electron is. So I give you 5 seconds. Okay, so phosphorylation is not right because phosphorylation means adding phosphate. Oxidation, yeah, oxidation is correct. Okay, oxidation is correct. Reduction means gain an electron. Now let's work on the section 3. Okay, so the catabolism. Uh, so catabolism means break down the macromolecule to the small molecule. Okay, so catabolism can release the energy, can make more small molecule for the anabolism. So the actually one catabolism break down the molecule so they can because they have to break down the chemical bonds. Okay, so they can release more electrons. Okay, electrons. So the electrons so those electrons carry energy. Okay, so electrons can be stored in the NAD or FAD. Okay, and so NAD FAD will be reduced. Okay, so they have reducing power. Okay, and then so the energy in the electron will be transferred and stored to the ATP. Okay, so we talk about this later. As what we can see, we have three major metabolism pathway. Okay, so one is the aerobic respiration. Second is anaerobic. The third is fermentation. Okay, so as what we can see for all these three. Okay, so the first one is same. Okay, so the glycolysis is same. So all these three pathways have the same first step. The first step is glycolysis. So glycolysis can produce two ATP and carbon dioxide, one molecule carbon dioxide and and electron. Okay, and then the three carbon unit. Okay, three carbon unit is the product of the glycolysis, can be used to Krebs cycle. So the second step is same in aerobic and anaerobic pathway. Okay. However, so the uh, fermentation doesn't have the step 2 and step 3. So the fermentation will use the 3 carbon unit to uh, to uh, to to accept the electron and release the energy. Okay, to produce alcohol and either alcohol or acid. Okay, so that's it. So it's done. However, for the aerobic and anaerobic, so the Krebs cycles can continue degrade the 3 carbon units. Okay, to break down the chemical bonds between the carbons to release the energy. Okay, release the electrons. Okay, electron will be added to the NAD and FAD, and then so the electron will be transferred to the uh, electron transport system. Okay, electron transport chain. Okay, so electron transport chain actually is the third step. Okay, so the third step is different for these two pathways. Okay, for the aerobic respiration, so it uses the oxygen to accept the electron to release ATP. However, for the anaerobic respiration, so it uses the other molecules such as sulfate, nitrate, or carbate, okay, to absorb uh, to accept the electrons to release the ATP other than the oxygen. Okay, so the outcome we can see. So the aerobic respiration can release most energy. Okay, can release the highest number of the highest energy. So it can produce 36, 38 ATP. However, for the anaerobic, so it can make only 2 or 36. So it's less than the aerobic. Fermentation, so the fermentation makes the lowest energy, so only 2 ATP. Now let's go through this pathway one by one. Okay. For aerobic respiration, actually, so there's a series of reactions to break down the glucose. Okay, to break down all the chemical bonds between the uh, carbons. Okay, to generate carbon dioxide and water and release all the energy between the carbons. Okay, so it has 3 steps. First is glycolysis. Second is Krebs. Third is the respiratory chain. Okay, so it uses the free oxygen to accept the electron and the hydrogen to produce water. Okay, to produce water and release energy. So it is widely used by many bacteria, fungi, protozoa, and animals, also humans. Okay. Anaerobic respiration. Um, so this pathway doesn't need the oxygen. Okay. So this pathway also has glycolysis and Krebs cycle. Okay. So third is same electron transport chain. Okay. However, so it doesn't use oxygen to uh, accept the hydrogen and electrons. So it uses nitrate sulfate carbonate to accept the electron and hydrogen. Fermentation actually is considered as incomplete oxidation, okay, because it has only the glycolysis, okay, so that's it, okay. So it cannot make more energy, okay. So the organic compounds will be used to accept the electron, okay. Actually, the organic compounds is made from the pyruvate, okay. Now let's talk about the glycolysis. So the glycolysis can produce 2 ATP, okay, 2 ATP, and so the glycolysis will break down the glucose. Glucose is 6 carbon sugar, okay. Break down the glucose to 3 carbon units. So that's pyruvate, okay, pyruvate. So it can produce 2 ATP. And release energy to be stored in the NAD, okay. Make it 2. So second is the Krebs cycle. So the Krebs cycle, so we'll use acetyl-CoA and then to break down the chemical bonds between the carbon atoms and release energy and release the carbon, okay, to form the carbon dioxide. So it can produce 6 NAD, okay. So it can release a lot of electrons. So those electrons can be stored in NAD and FAD. So it can make 6 NAD and 2 FAD, okay, and 2 ATPs, okay. So NAD and FAD can carry the electrons to the electron transport chain, okay, to form the energy. So we can see here, so the pyruvate will be conferred to the acetyl-CoA. Okay, acetyl-CoA will bind with the oxaloacetate. Okay, oxaloacetate is the 4 carbon unit. So they can bind together to form another 6 carbon unit. So this is called the citrate. Okay, citrate can continue be degraded to release the carbon. Okay, because so the bond chemical bond here will be cut. Okay, cut. Actually, so for example, these 2 chemical bonds will be cut and release. And so these 2 carbon will be released to form the carbon dioxide, okay. And then the citrate will become to the 4 carbon unit again. So this is oxaloacetate, okay. So it can release a lot of electron, and so this cycle can also release ATP, okay. Now let's talk about the respiration chain. So this is a major step to form to make the energy, okay. So this is the chain of special redox carrier, okay. So the chain units are embedded in the cytoplasmic membrane, okay, in the bacteria, okay. also in the eukaryote sites. So this is in the mitochondria. Okay, so you can receive the electron transport from NAD and FAD. Okay, once NAD and FAD upload the electron to this chain. Okay, so the electron will be released to the outside of the cellular membrane. Okay, that's mean in the outside space in the outside space in the space of outside the cellular membrane. So the hydrogen ion. So the electron concentration is very high. Okay, it's very acidic. Okay. So the uh, hydrogen ion and the electron will generate energy, and then so they will be accepted by the oxygen to make the water. So the compounds in the electron transport chain are the NAD dehydrogenase, flavoprotein, okay, coenzyme Q, okay, ubiquinone, okay, and the cytochromes. Cytochromes, so they contains the metal ions such as iron, okay. So these are the factors very important for the cytochrome function. So they can regulate the cytochrome to accept electron, okay, or either donate. So as what we can see in this diagram, so once NAD or FAD upload the hydrogen ion, and the electron, okay. So so the cytochrome, so these are the cytochrome units, so they can transfer the hydrogen ion and the electron to the space of outside of the membrane. Okay, so in this space, so the hydrogen ion concentration and electron concentration is very high, it's very acidic. Okay, so this is ATP synthase. So it's like a motor. Okay, so so there is a channel in the center of this synthesis. So the hydrogen will get into get in. So we'll get to inside of the cell, okay, of the cell, and push this synthesis spin at a very high speed. Once this synthesis is spinned at a very high speed, so you can convert ATP to ATP. So it can add the energy to the so it can make the ATP phosphorylated, okay, and another phosphate group to the ADP, and to form the phosphate bond, okay, to store the energy in the terminal phosphate group. So the hydrogen ion and the electron in the cytoplasm of the cell will be accepted by the free oxygen, okay, or the other compounds, okay. For the free oxygen, so they make the hydrogen, so they make the water, okay. For the other, they make the other compounds. So we talk about this later. So this is called aerobic respiration. So this is called anaerobic respiration. So for the eukaryotes, okay, eukaryotes actually eukaryotes make release energy in the mitochondria, okay. So this is the structure of mitochondria. So actually, so the cytochrome exists on the Cree state, okay, on the Cree state, okay, Cree state. So actually, so the hydrogen and the electron will be deposited to this space, okay, this space. So this space has very high hydrogen concentration, so you can diffuse to inside of the mitochondria through the ATP synthesis, and let the ATP synthesis spin at very high speed, and then to produce ATP. Okay, so this is the process. So for the aerobic respiration, so the whole process is called oxidative phosphorylation. Okay. Especially in the electron transport chain, okay, because so the oxidative means the combination of the so that means the free oxygen accepts the hydrogen and the electron, so this is called oxidative. Okay, phosphorylation, so that means synthesis of ATP. Okay, so ADP will be phosphorylated with adding another the third phosphate group. So this is called phosphorylation. So the chemical reactions on the electron transport chain is called oxidative phosphorylation. So each NAD can produce 3 ATP. Okay, 3 ATP. So the FAD can produce 2 ATP. One FAD can produce 2 ATP. Okay. So this is oxidative. Okay, oxidative for the aerobic respiration. So we can see hydrogen and the electron will be accepted by the free oxygen, okay, to make hydrogen. However, since aerobic respiration use the free oxygen, okay, so so all the free oxygen are not completely reduced. Okay, certain free oxygen will not accept the hydrogen and the electron. Okay, so the free oxygen actually they are considered as the free radical. Okay, for example, the superoxide ion. Okay, and the hydrogen peroxide. Actually, so they are toxic. Okay, so they are the free radicals. So that's why for all the animals and all the aerobic cells, so when they use oxygen to reduce to make the energy, actually, so the action can be toxic, so they can make the damage of the cells of the tissues. Okay, so that's why so we will get old. Okay, so they will continue damage our cells and the tissues. So that's why our cells has a lifespan. Okay, so they cannot live forever. However, so the cells have specific enzyme to detoxify of the free oxygen. Okay, so for the animals and humans, for the eukaryotes, so they have two enzymes. Okay, such as superoxide dismutase and catalase. Okay, however, for the microorganisms, okay, for example, the streptococcus. So they don't have this enzyme. Okay, however, they have peroxidase. Okay, peroxidase. So those enzymes can detoxify the free oxygen. Now let's talk about the the final step. Okay, final step of aerobic respiration. Actually, so this is anaerobic. Okay, anaerobic. So the anaerobic, they use the other chemicals, okay, the other compounds such as nitrate, okay, or the sulfate, okay. So for example, for the nitrate, so nitrate can accept the hydrogen and electron, okay, so this is made by the nitrate reductase, okay, to become nitrite, okay, nitrite and water, okay. So this is a very important process. So this is called denitrification. Okay, denitrification, because the nitrate can be continuously reduced to the nitric oxide and the nitrous oxide. Okay, and eventually, so they will release the nitrogen gas. Okay, so this is very important for the recycling of nitrogen in our Earth. Okay, for example, the species of Pseudomonas and Bacillus, so they have the specific enzyme to continue reduct the nitrates. Okay, so this means when these cells utilize the energy, so they will produce eventually produce the nitrogen gas. Okay, to recycle the nitrogen in our Earth. Okay. So the other compounds can be the carbonates and the sulfates. Okay, carbonates, sulfates. So they can also make the ATP. Now let's talk about the fermentation. Okay, fermentation. Actually, fermentation can only make the the last ATP, the last energy. Okay, so this is incomplete oxidation. Okay, incomplete oxidation. So this is not oxidation. Okay, this is not the real this is not the real oxidation. So they still use the glucose. Okay, still use the glucose. So this is very simple. So for example, so this is the diagram that shows the fermentation. So actually, so the first step is the same. It's also glycolysis. So it's also glycolysis. Okay, glycolysis actually makes the 3 carbon units, right? So the pyruvate. Okay, pyruvate can be either convert to the acid alcohol. Okay, or the lactic acid. Okay, so those also can be used to accept the hydrogen and the electron ion. Okay, hydrogen and the electron. So these are the two molecules for the reduction. Okay, and once they accept this the hydrogen ion and the electron, so the energy will be released. Okay, so the final products will be the either alcohol or the lactate. Okay. uh, some other microorganisms can also produce the butanol and propionic acid. Okay. So the final products can be used by a human. Okay, that's why we use those type of microorganism to make the products that we need. For example, the the alcohol. Okay, ethanol. Okay, so we can make the wine, make the beers. Okay. We also use the fermenters to make acetone, butanol. Okay, so those are used as the solvent. Okay, or the acid, organic acid like acetic acid, like a vinegar, right? Vinegar and lactic acid, vitamins, antibiotics, and hormones. Okay, uh, so we also use fermentation to make those. Catabolism of the compounds. Okay, um, so the specific enzyme can catabolize the macromolecules such as the polysaccharide to the glucose to the 6 carbon glucose. Okay, lipid can be broken by the lipids to the glycerol and fatty acid, right? So the fatty acid can be converted to the 3 carbon units by the beta oxidation, and the glycerol will convert to the dihydroxyacetone and dihydroxyacetone phosphate, then go to the glycolysis to become the 3 carbon units, then get into the Krebs cycles, okay, to release energy. So for the protein will be break down to the amino acid by the protease. Then amino acid can be removed. So the animal group will be removed by the deamination, okay, deaminase, okay, and then so the remaining part will be converted to the pyruvate and then degraded by the Krebs cycles. Now let's work on this question. What is the maximum net yield of ATP per molecule of glucose for each of the following type of respiration? So I gave you 5 seconds. So the correct answer is A, okay, aerobic respiration. So this respiration makes the most ATP. Now let's talk about the efficiency of the sear. So the catabolism makes the small molecules, so those are the metabolites, okay? So they can be used by the anabolism. Actually, one molecule can serve multiple purpose process, okay? So they can be either for the nutrients and used by the cells. Okay, actually, so the catabolism and the anabolism are linked together. So the ability for the microorganism to manage the catabolism and the anabolism is called amphibolism. Okay, as what we can see in this diagram. Okay, so we can see all these nutrients actually, so they can be degraded to the basic units. Okay, the basic units can be for these 3 basic units. Okay, amino acids, sugars, and fatty acids. So they can be transferred to the pyruvic acid and acetyl CoA. So those are the basic molecular or the metabolites. So they are used to either for the energy releasing or for the anabolism to build another nutrients such as the amino acid or the fatty acid or another glucose. Okay, so the pyruvate can be made to amino acids or the sugar. Okay, acetyl CoA can also be used to make the fatty acid. So anabolism actually is the process to make macromolecules, okay, such as the amino acids, fatty acids, uh, nitrogen base. Actually, nitrogen base is for the nucleic acid, is to make either DNA or RNA, okay. Vitamins, sugars, okay. So the carbohydrates biosynthesis. So the glucose has a role in the metabolism and energy use. Okay. So the glucose will be so the first step for for the metabolism of glucose is glycolysis to form the glucose 6 phosphate. Okay, uh, and then so so this one can be used to form another can be used to form a glycogen. Okay, so it can also be used to form the peptidoglycan. Okay, so it can also be used to form. So it can also form the ribose. Ribose. Okay, ribose is for the nucleic acid. Polysaccharides. Okay, polysaccharides. Polysaccharides is the polymer of the glucose. So the proteins actually there are 20 animal acids. So the proteins are the polymers of animal acids, right? So the proteins are used to make nearly all the cellular organelles, such as membrane, uh, cellular wall, cellular organelles. Okay. For example, the E. coli. Okay, E. coli can make all the 20 animal acids. However, our human cannot synthesize all these 20 amino acids. So that's why. So in our colons, because in our colons there are tons of E. coli. Okay, so those E. coli uh, can be benefit uh, to the human. Okay, because E. coli can make the can make a can make the vitamin K. Okay, because our human cannot synthesize vitamin K. So we have to so once E. coli synthesizes vitamin K, so our colon can absorb those vitamin K for human to use. Okay. So the nucleic acid actually, so it is the DNA and RNA, right? So their function is for the hereditary process, like so they store all the hereditary information, and the cells can use this information to produce the protein. Okay. So assembly cells. So once the cells divide, okay, so the anabolism and the catabolism work together to make a two copy of the nucleic acid and organelles. Okay, and then so these two copies can be distributed to daughter cells. So Now let's work on this question. The ability of a cell to integrate molecular using and molecular building pathways to improve cell efficiency is known as. I give you 5 seconds. The correct answer is amphibolism. Okay, amphibolism. So now I have covered all the contents of this chapter. So this is the appendix. So I recommend you guys to review the contents by using this appendix.